Hi everyone from my side. I apologize for the technical inconveniences and for me not being there, but I spent the last 4 days in the hospital, so it's all been hectic. The topic of my today's talk is a specific organelle in our cells called the mitochondria, and how they interact and what they do with cannabinoids, or actually more in fact how cannabinoids affect these little tiny organelles. So first, let me explain a little bit what mitochondria are. They look something like this, like spherical little organelles, and we can imagine them as being the power plants inside of our cells. So everything we eat and a majority of oxygen that we breathe in, they all go to these tiny organelles where they're transformed to these, to the energy that our body and our cells understand. And this is a very specific molecule. So these organelles are very important for the functioning of our body, and they're very well known in medicine. They're connected with very diverse range of symptoms and diverse diseases. And interestingly enough, it has been shown that they're also very important in the biological aging of our body. The symptoms when these tiny organelles, mitochondria, are not functioning can be very, very different. Sometimes they show up as neurological symptoms. Sometimes we see different kinds of weakness in the muscles, or in a weaker function of the livers or the kidneys. It depends in which tissues or in which organ these organelles are not functioning properly. In medicine, we know something that we call the primary mitochondrial diseases. These are cases where the mitochondria themselves have a mutation, and they have, therefore, of course, they don't function properly, and we see different kinds of symptoms. And then there are secondary mitochondrial disorders, where the mitochondria, the DNA of the mitochondria is intact, but their function is somehow, for some reason, impaired. And today we know that these secondary mitochondrial disorders are many times associated with lifestyles, and sometimes we can correct them with changing lifestyles, choosing more appropriate diets, working out, and different kinds of lifestyle intervention have an important role in these secondary mitochondrial disorders. Now, mitochondria are present in all cell types and all throughout our body, and our body is quite an interesting construct. We have different kinds of systems working in our body, from our bones to our neurons, from the lymph system, cardiovascular system, and of course, we know that we have very different set of organs. But when we look at all of these organs under a microscope, we see an array of very different cells. When we look at, for example, a bone cell, it looks drastically different from a pancreas cell, or a brain cell looks very different from a kidney cell. But so these would be, for example, a group of immune cells. This would be a group of muscle cells, blood cells. But when we would look at a single cell, how they function, they look something like this, like a tiny ball, and very specific organelles inside of it. And then these tiny organelles, these ones are called mitochondria, and these are the ones that we will give special attention to today. Now, this is a video showing what it would look like if we would zoom in with a camera into our body. These little tiny spherical things, these are our cells. And if we zoom into a single cell, we see that we have quite a complex structure. This blue thing is where all of our genes are. This is endoplasmic reticulum. And these brown things, these are the mitochondria. And these are the ones that can be found only a few in a cell, or also up to a few thousand mitochondria, depending on how much energy a cell needs. Now, if we imagine that we have quite many millions and trillions of cells in our body, and they all do the same things, it's very important to keep in mind that all of our cells, for our body to function, have to communicate with each other. And actually, our luck is that our cells are very communicative. They're very talkative, and they're also very good listeners. And cells constantly send each other messages or texts and exchange information of what's going on in one cell and what the other needs, and all of this communication is happening quite at a rapid rate. Of course, cells are limited, that they cannot text each other SMS messages or WhatsApp messages, but they have a different view. They synthesize different kind of molecules. We call them signaling molecules, with which they send these messages and tell what's going on. And these signaling, we know a huge array of signaling molecules. For example, hormones are also signaling molecules, and cannabinoids, of course, are also signaling molecules. And cannabinoids are very well known as signaling molecules that help carry this message from one cell to the other cell. And in these signaling cascades, we know that, of course, one cell is sending the message, and we have to have a cell that has an appropriate receptor to hear this message, or to be able to respond to this message. And in our body, a vast majority of cell types has cannabinoid receptors. So majority of cells in our body can hear the message that cannabinoids are sending. But this aspect of the signaling is quite well known. Of course, we have different types of receptors that respond to these cannabinoid messages, and the CB1 and the CB2 receptor are already very well known, and CB3, that was formerly known as the GPR55, is also an important signaling receptor for these, for cannabinoids. And then we have a whole TRPV channel of receptors that are also quite sensitive to receptors, to cannabinoids, and many other types of receptors as well. So this role of cannabinoids as basic signaling molecules inside of our body is not a new one. A more kind of a new and less well-known approach is that we have to understand that not only do cells communicate one cell to another, but also inside of a single cell. If you remember how many different organelles are in one single cell, a very efficient communication has to be happening inside of a single cell as well. And this is what we call intracellular communication. So how well the nucleus is telling other organelles what is happening, how well the mitochondria know how much energy they have to produce, and all of these processes have to be very well regulated, and this happens through intracellular communications. So the communication inside of one single cell. Especially important aspect is, it seems that how well mitochondria talk or exchange information with the endoplasmatic reticulum and also with the cell nucleus. And depending on how well these organelles interact, how well they exchange information, we now know that if this is flowing smoothly, the body is, cells and the body and the tissues are functioning properly. But if there is something wrong in this communication, a very diverse array of symptoms and pathologies can happen. We know now that, for example, insulin resistance, different kind of cardiac situations or pathologies, and Alzheimer's disease are already proven to have an element in their progression of this dysfunction of this communication inside of a single cell. One interesting way how the nucleus and the mitochondria talk to each other is through a molecule called NAD. And very well-known researcher of aging, Dr. Sinclair, has been studying this in great detail, and he showed that young cells always have a very high concentration of this NAD molecule, and this is how this is a molecule of how the nucleus tells the mitochondria how much energy this cell needs. And he showed very efficiently that young cells have a lot of this communication going on, so the mitochondria always know how much energy it needs, and the nucleus always knows what's going on outside of the cell. And in old cell, this communication is, this molecule is in much lower concentration, and this communication is not flowing so freely and so effectively anymore. In a very cute way, he compared this young cells with a freshly married couple or a freshly in love couple. They talk to each other all the time, they spend a lot of time with each other, they tell each other everything from what they want 10 years in the future to what they're wearing, to what they're thinking, everything. This is a young cell. In an old cell, this is a couple that's been married 25 years, and they can't bother to tell each other anything anymore because no one really seems interested anymore. And this symbolizes the level of communication, the level of information exchange that's happening between the nucleus and the mitochondria. So in young cells, a lot of this is happening, and in old cells, very little. And he actually did an experiment in mice where they injected this molecule NAD in older mice, and they noticed that very soon started to regenerate, and their body and the way they behaved was of young mice. And when they looked at their internal organs also became rejuvenated and under a microscope looked like much younger mice than they actually were. So there seems to be a very important correlation between this communication and biological aging. If we would to translate this experiment on mice to humans, they said that they were able to rejuvenate someone who would be in human age 60 years old to a 30-year-old one. So this seems to be a very important factor in the overall process of biological aging. Now this is another animation to show you how how mitochondria actually, just a second, oh sorry, there's something. How mitochondria are actually where in the cell they are and what they do. So this would be a cell, and this tiny thing is a mitochondria, and this cell has rather few. And if we take a closer look at the mitochondria, these little red dots is the energy that our body and our cells understand. It's called ATP, and all of the food we eat has to get converted into mitochondria in these ATP molecules. And the more of the ATP mitochondria produces, the more energy our body, our cells, our organs has to use. And if we take a look at inside of one of these organelles, we see that this organelle has two membranes, an outer membrane that's around, and then this one that is kind of has these cristae or this invigorations inside, and this actually on the inner membrane is where all of this transformation of food to energy is actually happening. Okay. And the mitochondria are actually there's a hypothesis of how these mitochondria came in evolution, how they came into our cells. And the most well-accepted hypothesis is that the mitochondria were actually used to be long time ago in evolution, bacteria that kind of became symbiotic with one-cellular organisms, and they found that the symbiosis to be very efficient. That is how they stayed. This is how multicellular organisms could develop. And one important point that points in this theory is also this, that the mitochondria are actually the only organelles that have their own DNA. So they carry their own genes with them. When we look at inside of the mitochondria, we can find very specific mitochondrial DNA. It only carries 37 genes, but 37 very important genes. If we were to compare this with all of the genes that the nucleus is carrying, the nucleus would be like an encyclopedia, and the mitochondrial genes would be a tiny leaflet. So it carries a lot less information, but very, very vital information. And we know now that if mutations happen in this mitochondrial DNA, it carries very significant consequences for the health and the disease formation in humans. Another important point is how, of course, from the medical point of view, is how these mitochondria are being inherited. And we know that vast majority of the mitochondria are not in the head of the sperm, and when the sperm enters the egg, this part where the mitochondria are is lost. So the offspring always, in humans, and the offspring always carries maternal mitochondria. So this is also very evident when we see it how mitochondrial mutations are inherited. We always see that they're inherited over the maternal genetic line, not the fathers, but always the mothers carry on mutations in the mitochondria. Since we know that mitochondria are how well the mitochondria function and how well, if they have any mutations and how good they function, is not only directly linked to health and disease, but is also very important in aging, then the joke that if you want to know how your wife is going to look like in 30 years, take a look at her mother is actually not so far-fetched. But okay, all the jokes aside, the mitochondria have very important roles to play. So first of all, of course, I already mentioned that they're important in producing energy for the functioning of our body. They're also very important in calcium homeostasis, so how well the cells are, how this calcium signaling is happening. Then they're very important in apoptosis. This means a programmed cell death, when cells know when it's time for them to die and be replaced by new cells. Because there's so much metabolism going on in the mitochondria, a lot of radical oxygen species are also created. So it's very important that this balance between how much reactive oxygen species are created and how much antioxidants are present to scavenge this is also very important. They are known to kind of be the ones that are orchestrating all of the metabolism in the cell. Steroid biosynthesis, other hormone biosynthesis, it has a huge array of important roles to play. And now we come to the part where we ask ourselves, so what do cannabinoids do to these tiny organelles, or do they have any kind of function? Now, the first surprising part was that when these organelles were studied, they found that on this outer cell membrane, we actually have CB1 receptors. So this is the receptor that is activated upon anandamide or THC or some other cannabinoids, is actually present on the outer surface of these organelles. Now this was first very surprising, but it is now proved beyond doubt that these that the mitochondria have CB1 receptor. And so far CB2 receptors have not yet been found, but it was found that when on the outside of a cell a cannabinoid docks onto the CB2 receptor, the reaction that happens inside of the cell travels into the mitochondria, releases calcium, and this is actually what causes all further reaction. So if we don't have functional, well-functioning mitochondria, even if we have cannabinoids, this signal inside of the cell will not happen the way it needs to happen if the mitochondria are not functioning properly. And we see that this is a very important part in what cannabinoids do or how they do it, and why sometimes we see a very different response in different individuals with the same cannabinoid treatment. So this is just a schematic depiction of how we have these cannabinoid receptors, CB1, on the outside of the membrane, and how this reaction influences the mitochondria itself. Now, this is another very short movie showing so the membranes outside of our cells and outside of the mitochondria is such a tiny small lipid bilayer. So we have two layers of lipids, and this is on the cell outside. This surface is not rigid. We can imagine it as very flowing. And one of the important factors that was shown that what cannabinoids do is that they affect how fluid or how rigid these membranes are. And because we know that you see this movement, and because we know this membrane is lipid, and we know cannabinoids are also lipid soluble, they're fat soluble, they can pass through the membrane even if we don't have receptors on the cell surface. They can get into the cell and affect the mitochondria from there. And now if we take a look at what do cannabinoids actually do to the activity or to the function of the mitochondria, what was shown was that vast majority of cannabinoids, meaning endocannabinoids like anandamide, or phytocannabinoids such as THC or CBD, they affect mitochondria in a way that they reduce the amount of ATP that the mitochondria produce. So they actually reduce the activity of mitochondria. So we have seemingly less available energy. But what this actually means for the functioning of the body is very much dependent on what cell types we look at. And Dr. Chandel showed this very effectively in his studies. If we take cancer cell and we reduce the amount of available ATP, this means less proliferation and more survival. So cancer cells are known to need a lot of energy because they divide so fast. And if the activation, or if we reduce the activity of mitochondria in cancer cells, we can cause slower cancer growth, or we can actually cause shrinking of cancer and death of cancer cells because they don't have enough available energy. The story is very different if we look at stem cells. When we change the activity of mitochondria in a stem cell, we can see that the stem cells will start to differentiate. And if we take T cells, which are immune cells, and reduce the activity of the mitochondria, we can see that they can change their function. And this again can be good or it can be bad, depending of course on what do we want to do with the immune system, how do we want to modulate it. But usually when we're talking about pathological conditions, then modulation of the activity of mitochondria can actually be very beneficial. As I mentioned, there's a lot of activities happening inside of the mitochondria, and these ROS stands for reactive oxygen species, and there's a lot of them created in the mitochondria, and if there is too much reactive oxygen species, this can cause changes in either they can even cause mutations, or it can change the activity of mitochondria. And all of this, if it's out of balance, that there's way too much reactive oxygen species, then the body can scavenge. This can lead to to development of tumors. And this is another important point where cannabinoids can do their role, because a lot of phytocannabinoids are known to be antioxidants. And if we have enough antioxidants with a lot of reactive oxygen species, and if this is imbalanced, the mitochondria can keep up their important function in the body. Now, this is a representation of a whole cell, so this is one cell. And if we look at different receptors on the top of the cell, we can see that most of the signals that are happening, that are coming into the cell, they flow over calcium. This what happens with the level of the calcium inside of a cell is very important. That is why the cell is always aiming at keeping the level of calcium very low. And when an activity happens or a signal comes, we have these peaks of calcium, and then they return back. A peak of calcium, and then they return back. Changing the level of calcium is also a way of how cells communicate to each other. And because this is quite difficult to imagine how this is happening, I have a very short movie showing. So this for example is a single cell, and this is a single cell, and you will see with the bright orange color how this information of calcium is traveling from one cell to the other. So this is how this cell here sent a signal of calcium to other cell, because calcium is one of the signaling molecules that cells used to talk to each other. One important model in where the importance of these calcium concentrations and these fluctuations was shown was in a part of the brain we call hippocampus, and it was shown that this level of calcium and how it spikes and falls is very important in seizures. And when in models we induce that the calcium is jumping up and down and jumping up and down and not coming back to a steady level, this is usually associated with following seizures, either epileptic or other kinds of seizures. And on such a model where they induced these seizures, CBD was used to show if CBD could affect these spikes and these typical epileptiform activity we see of the calcium. And it was very interesting that if they first induced these changes, that would lead to a seizure, and then applied CBD, what happened is that this epileptiform activity was not happening anymore with the CBD, and that it came back to a basal level. This would mean, of course, that even if there was a trigger for epileptic seizure, it would probably be drastically reduced by CBD. And on the other hand, if they first gave neurons CBD and then they tried to induce these calcium spikes, this was even further drastically reduced, and this is how we can explain by looking at the calcium signaling is one of the ways we can explain, or probably one of the pieces of puzzles we can look at when we try to explain why CBD is in many cases very effective with epileptic and other kinds of seizures. So we see that it significantly affects how this epileptiform activity in neurons is happening. And then another important fact is that this is what we see many times with cannabinoids. If we take a normal cell and we give it cannabinoids, we usually don't see much changes. But if we take a cell that is way out of its biochemical balance and apply cannabinoids, we can see drastic changes. And with neurons, the same thing happened. So if they took neurons that were normally excited and gave them CBD, they saw that there's a little bit of increase in calcium signaling, but nothing much. But if they took highly excited cells, neuron cells, so that would mean cells under a lot of, with a huge amount of excitability and a huge amount of activity, to such neurons and gave them CBD, the neurons started to come back to biochemical balance. They weren't so highly excited anymore and didn't produce so much neurotransmitters anymore. And this is also a very important fact, because we know that in neurons, very high levels of excitability can cause death of neurons or other kinds of symptoms. So this activity of the cannabinoids, of the CBD in this case, to reduce this excitability is very important for the general protection of the neuronal system. And then in this experiment, it was shown that this calcium that we see inside of the cells changing, that this calcium actually comes from the mitochondria. So the mitochondria have to cooperate in this role very efficiently, so that this calcium signaling can happen normally in the cell. And so it has to be very tightly regulated. Once again, the whole cell has to communicate very well with the mitochondria for this to happen in a good way. And another very important part, or another important element that was shown for cannabinoids and CBD in particularly, is that CBD has a very strong protective role on mitochondria. So if they expose mitochondria to chemicals, toxins, other molecules that would disrupt how the mitochondria are doing their role of producing energy, if they had CBD present, the mitochondria were able to maintain their role even if there were chemicals around. If the CBD wasn't there, the mitochondria would either die or have a drastically reduced activity. But if enough CBD was present, they could do their function practically normally, even if the conditions weren't ideal, even if there were toxins around. So this role of CBD or also other cannabinoids seems to be very important in their health effects. Okay, so this is now just to kind of close up. This is an image of a cell and the cannabinoid receptor, and we know that cannabinoids can either enter directly and affect the mitochondria, or they can go through a receptor, but also in the end affecting the levels of calcium, and the mitochondria have to be involved if the cell wants to respond to this signal of the cannabinoids, either with autophagy or apoptosis. Whatever the cell wants to do in response to a cannabinoid, it has to have a well-functioning mitochondria. Now this is just to give you an overlap. We now know that the closer we look, the more ways we find how cannabinoids have this very amazing, very complex role in our body, in our organs, tissues, in cells. Even if we look at the single cell, we see a very high complexity. And when scientists have no idea how to explain all of this complexity anymore, they usually take that and give it a name, something om. So when scientists were studying the beneficial bacteria in our gut, they didn't know how to explain all of this complexity, and they called it microbiome. And the same thing happened now in the field of cannabinoids as well, because this endocannabinoid system is extremely complex. We now call it, there's a new name for it, and we call it the endocannabinoidome. And this explains or takes into account all of the targets we know, all of the receptors. We now know that we have over 100 endocannabinoids in our body, that there's way more than just 2 receptors, that the receptors are present not only on cells but also inside of the cell. And all of this complexity can be, we just need to keep in mind that it's all, the more we study, the more complexity we see. But even with all of this complexity, the plants seem to have it all figured out, because the sum of all of the plant cannabinoids, terpenes, and all of the 1,000+ molecules that cannabis sativa actually has kind of plays in very nicely with the complexity of our endocannabinoid system. And the aim of all of these, either our own endocannabinoids or the phytocannabinoids we take in, is to ensure that our body from the cellular level on can function in the best homeostasis possible. So this is not a static situation. A lot is happening in our body and in every cell, but that this kind of is kept in balance. Homeostasis actually means that it's a dynamic balance, that we don't jump very far out of our ideal biochemistry or our ideal situation in our body. And cannabinoids seem to be this primary SOS signaling molecule that tells our body to return back to its homeostasis, back to its core biochemical balance. And on the one side, of course, we see huge complexities, and I'm very happy that there are so many studies new coming, and we see even more complexity with new studies, but on the other hand, we have a very simple answer as well. So, as far as the science can sound very complicating, the nature sometimes has very simple solutions. And to close up with Dr. Meshulam's saying, with which I agree very strongly, is that this plant, or its cannabinoids, it's all array of chemicals it's carrying. It seems to be that it's still a very neglected treasure chest for pharmacological action. Okay, so thank you very much, and I am open for questions.
Hvala tudi tebi, Tanja. Nekoliko nas čas priganja, tako da žal ne bi postavili vprašanj. Eno vprašanje. Čak samo trenutek.
Dober dan, Tanja Bagar. Vemo, da ste napisala knjigo v nemščini. Kdaj lahko pričakujemo knjigo v slovenščini? Hvala.
Hvala, da ste namesto mene postavili to vprašanje.
Kdaj lahko pričakujemo knjigo v slovenščini?
Ja.
Tega še ta trenutek ne morem povedati. Veliko prej bo v angleščini, to je že zelo v pripravi in v delu. Za slovenščino pa ne vem, ena možnost se kaže in upam, da bo nekje, morda v drugi polovici naslednjega leta, če bo vse šlo po planih.
Upam, da že ne bo zastarela do takrat.
Ne bo.
Saj vse updatamo.
Dobro, prav.
Tanja, najlepša ti hvala.
Z veseljem.
Hvala. Adijo. Hvala.